everyone. My name is Danielle Gatti Palumbo, and today we'll be presenting on the Journal Club of Deucravacitinib in active psoriatic arthritis, the efficacy and safety of up to 52 weeks data from the randomized placebo double-blind POETYK PsA-2 trial. So first off, what is deucravacitinib? And deucravacitinib is a TYK2 inhibitor, and tyrosine kinase 2, or TYK2, is an intracellular kinase enzyme that is a member of the JAK family, and this mediates the cytokine signaling downstream of type 1 interferon, IL-2, and IL-23, which are involved in the pathogenesis of psoriatic arthritis. So TYK2 inhibitors inhibit this downstream signaling of these cytokines, and TYK2 specifically is highly selective. It selectively binds to the regulatory domain, unlike other JAK inhibitors that bind to the catalytic domain and target a broader immune pathway, causing more immunosuppression and side effects. TYK2, because of its more selective, it is more selective to a distinct immune pathway. It has minimal impact on other JAK inhibitors and therefore has less immunodeficiency and side effects. So looking at our TYK2 deucravacitinib, it is currently FDA approved for psoriasis and psoriatic arthritis. And in this study, this was a multi-center randomized double-blind placebo-controlled phase 3 study of a placebo crossover design looking at the efficacy and safety of deucravacitinib in active psoriatic arthritis. Patients in this study needed to be both biologic-naive, or they could have previously tried less than 2 TNF inhibitors or had tried a 1 NSAID and or conventional DMARD and or 1 TNF inhibitor. There was also included an apremilast safety reference arm, and patients that were enrolled in the study needed to be adults with active psoriatic arthritis, active arthritis defined as more than 3 tender or swollen joints, high sensitivity CRP, or active or history of psoriasis. Patients were excluded if they had current inflammatory joint disease other than psoriatic arthritis. Patients in this study, there were 729 patients that were enrolled and randomized, and they were randomized at a 3:3:1 in deucravacitinib 6 milligrams daily placebo group that continued up until week 16, and then they crossed over into the deucravacitinib 6 milligram dosing arm, or apremilast 30 milligrams twice a day. And those that completed the 52 weeks can then enroll into an open label extension, and randomization was stratified by previous TNF inhibitor, their previous conventional DMARD at baseline, or high sensitivity CRP. So in statistics wise, a sample size of 700 patients was estimated to achieve a 99% power for the ACR 20 response at week 16 and for all secondary endpoints. They also used pool analysis from the POETYK PsA-1 trial to ensure sufficient power to detect efficacy and also control for that type 1 error. A two-sided alpha level of 0.5 was used for all efficacy and analysis. The Cochrane-Mansell-Hansel test was used to stratify with the previous either conventional DMARD at baseline or TNF exposure or CRP. The Klopper-Pearson estimate was used to evaluate the 95% confidence interval, and continuous endpoints were analyzed with the ANOVA. Any patient that was missing data was considered a non-responder, and all secondary endpoints were analyzed with the pre-specified hierarchical testing procedure to control for overall type 1 error. So looking at the results of this journal and clinical trial, there was 729 patients that were randomized, so it met its power. And the baseline characteristics were pretty much well-balanced. About 60% had been on a conventional DMARD previously, majority of them being methotrexate. 30% had high CRP, and 13% had been treated with the TNF inhibitor before. Looking at the primary endpoint of achieving that ACR 20 at week 16, this was significantly higher in the deucravacitinib group versus placebo at 54.2%. Versus 39.4%. And this significant difference was seen as early as 8 weeks and then continued through week 52. Similar findings were also found in the ACR 50 and also ACR 70. And then looking at the 8 ranked secondary endpoints, deucravacitinib also achieved statistical significant improvement in PASI SF36 and also achieved minimum disease activity. Other significant resolution in dactylitis was also seen in the deucravacitinib group and improvements in the DAS28 CRP. And then in the non-ranked secondary outcomes, the deucravacitinib had significantly achieved enthesitis resolution here in this part. And then looking at the 52-week data, the long-term efficacy, there was a continuous improvement seen in deucravacitinib through week 52. Even in those that were treated with placebo and then converted to the deucravacitinib group had achieved a rapid and sustained improvement similar to those that were initially in the deucravacitinib group. And then also ACR responses were consistent regardless of what they were on before, whether they were on an anti-TNF or whichever conventional DMARD they were on, and even baseline characteristics. Overall, deucravacitinib had a great safety profile. Majority of the side effects were mild to moderate and even self-limiting. Most of the common side effects that were with the deucravacitinib were COVID or URI or even some mouth ulcers. There was a higher discontinuation rate in the apremilast group, and that was mostly due to side effects. And the most common reason for any discontinuation with deucravacitinib was actually due to drug hypersensitivity or skin reaction. But overall, with safety, there was no new safety signals for MACE, VTE, malignancy, or opportunistic infections that we often will see with our JAK inhibitors. And so that was reassuring for especially deucravacitinib, and also gives light to its mechanism of action being more specific. So some strengths of this journal is that it was a large randomized double-blind placebo control, especially with that placebo crossover trial. It had comprehensive endpoints covering multiple manifestations of psoriatic arthritis, which we often will see in our patients. They don't present with just one feature. So this had efficacy with enthesitis, with dactylitis, with skin and joints. It included also patients that had failed previous therapies, including conventional DMARDs and biologics, which makes this a little bit more transparent for our patients, our real-world patients that we see in our office. And the 52-week data is a nice long-term insight into the efficacy and safety of this medication. Some limitations of this study was that anytime you study JAK inhibitors, you're always going to have that extra scrutiny and post-market surveillance for real-world data with JAK inhibitors. They're always going to monitor for the MACE, the VTE, and malignancy. And that's going to be just the landmark for any JAK inhibitor. The study was a little bit on the smaller side for those that had failed TNF. I think it was only about 13%. The placebo portion was also a short period of only 16 weeks. And the baseline characteristics, although they were well balanced, majority were white and 45 years old. So limiting our real world patient population. And then the use of apremilast as a safety comparison, even though there was a large discontinuation rate with apremilast, it was mostly due to a lot of side effects, and it has a completely different mechanism of action. So it's not a true comparison per se. So clinically, bottom line, deucravacitinib is a great option for our patients. It's an oral option. It has a great efficacy across multiple different domains of our PsA manifestations, and its selectivity for TYK2 also improves its safety profile and also has less immunogenicity as well as immunosuppression. So looking at this, I think this is a great option that we have in our patients, and overall it was a great study. So for more content on rheumatology and more journal clubs like this, please visit our RhAPP content room or our RhAPP PsA app. Thank you.